So the last stage of the electron transport system, or the last part or piece of the puzzle, is the ATP synthase complex, which was on our previous diagram, but we never really dealt with it. We had it there just so we understood that it was part of the process, but we never really did much with it. So what I'm going to do is, without drawing all of the other things, I'm just going to draw a little membrane picture here. So we'll slide down. Okay. So, little membrane. And the ATP synthase. I'm going to draw it in a little bit of a more complicated looking form. It essentially is a protein, but believe it or not, it's a protein with a very specific shape, and it looks, when we get it down to the electron microscope and actually get some views of this protein, it actually resembles an electric motor. It has the same sort of parts or components as an electric motor. And in fact, I don't know if it's in my textbook or your textbook, but one of them actually refers to those components like an electric motor. Maybe it's yours. Let me just look that up. Okay, so I just pulled up this picture to show you what we're about to draw and how complicated it really is. Now remember, it's a protein molecule. So it's made of amino acids in chains that are spirally twisted up and then are folded on each other by disulfide bridges and have other proteins associated with them in quaternary structure. And they produce this rather cool looking, crazy protein shaped thing. And so what you'll notice is it has this section embedded in the membrane, which is like a rotor, and it actually turns every time H plus is moving through it. H plus is a positive ion, so it's like a current. And so essentially what happens is the H plus moves through this rotor section of the protein, and it becomes mechanical energy. Just like an electric motor uses a moving electron to produce a spinning motion, right? This does exactly the same thing. It converts the moving current of an H plus ion, the moving charge, it converts it into a mechanical energy that literally rotates or spins. Then you have a little stalk down here below, and this large section on the bottom, which is called that says the catalytic head right there. This is the part, as it turns, that rotational energy that it has is then converted into energy for the production of ATP. So it's very much like a little electric motor. It's an electric ATP making machine. And it's quite cool. Except instead of wires plugged into it, it sits on a membrane, and the electricity, right, the charge, flows through as hydrogen plus ions, positive charge flow. So it's quite cool how that works. So we're just going to draw sort of a simpler representation of this. Okay? And then it's got this other little guy, sort of, my circles are getting a little bit crazy, this weird blue and brown thing on a stick. That's called the stator. And of course, that's what sort of attaches the two. We're not going to get into the complexities of electric motors, but if any of you are interested in that, you can read up on that, and you can find that it's basically the same thing. So we're going to draw this. We'll try to draw a little bit of that as much as we can, but of course, our picture won't be quite so complicated. So let me get rid of that. I think it's easier if I do it this way. There we go. So back to our drawing. Okay. So they have all these different colors, but we're not going to use different colors. We're just going to try to draw. I'm going to draw a central sort of structure like this. And then I'm going to draw another one that's sort of a little higher, and then a little higher. And I'll do the same on the other side. A little bit higher. And then we have to sort of do around the back. So around the back, we would have something like that. Okay? So it's a circular shaped sort of tube-like structure. And then We have what's called the sort of the shaft here that comes out of the bottom. Okay, and then around the shaft is this other part, which is, I'm going to sort of overlap it now onto the shaft. And it's sort of the same thing, but the shaft is in the middle. So we want the shaft, we want it to look like the shaft is actually going inside there. Something like that. Use your artistic ability to figure that out. Okay? And perhaps if I also just kind of shade these in a bit, just generally give the picture some depth. I feel like Bob Ross again. And we'll color in this little shaft here. Maybe yours is not the same color as mine, but in your world it can be whatever you want. There we go. We just color that in like that. It looks great. Wonderful. And then we can fix our membrane, bring it right up here, attach it, put some membrane on the other side, just like that. And we can't forget about our little phospholipid tails. So we'll throw some of those in there. We'll throw some of those in there. If I take a little yellow blob and I stick it right there, some of you will remember what that is. What is it? It's cholesterol in the membrane. We don't have to put that in. I just did that as a little fun thing, you know, to let you catch up with me as you're drawing, because I think you might be drawing a little slower than me. So there's our membrane with the ATP synthase complex in it. Now remember, the ATP synthase, what's happened previously on electron transport, the complexes, right? I wish I could do like a recap. If I had really good video skills, I could do that, but I'm just a teacher. I'm not a video editor, so I can't. But what's happened is that, let me see, what's a good color? I guess red. There's a whole bunch of H+ that has been pumped into the intermembrane space. Remember, we call this up on top here the intermembrane space. Intermembrane space. And then down below is the mitochondrial matrix. So we're talking about the intermembrane space is the space between the two membranes of the mitochondria. And the matrix is sort of the deep inside part of the mitochondria. And so all of those NADs and FADH or FADH2s were used up, the energy within them that came from, of course, the glucose molecule originally, because it came through glycolysis, turned into pyruvate, then acetyl-CoA as it went across the membrane, and then the citric acid cycle, and all that stuff happened and produced all this NAD and FADH2, which are now carrying the energy that used to be in the big glucose molecule. And the glucose molecule itself is gone, right? We've broken it apart, broken it up, stage by stage, step by step, and basically turned it into carbon dioxide and water. And that's all that's left of it. That's the other stuff that comes out. And then the glucose molecule has disappeared. So you can think about all of the order in the low entropy glucose molecule, low entropy, high energy, has been turned into the disorder of a bunch of little CO2 molecules, which are far simpler. It's like we broke the Lego apart into a pile of Legos. Less order, right? So higher entropy, but less energy. Where did that energy go? Into the FADH2 and the NADH and a few ATPs along the way. And now those FADs and NADs have worked very hard to transfer their energy through that complex electron chain, transfer chain, to create a very large or a substantially large hydrogen ion gradient across this membrane. Now the gradient doesn't just keep building up and building up and building up, because what's happening now is the hydrogen ions are going to flow through and out of the ATP synthase complex. Essentially, the ATP synthase complex is a little electric motor, And H plus is a moving charge. It's an ion with charge. In chemistry, electrical things, like even your nerve impulses, they're not zaps of lightning that you see or electrical sparks. What they are is the movement of electrical charges, which is still by definition electricity. So this creates a hydrogen current, a positive charge current. That's what that red line is. It's a current of positive charge. When you put a positive charge current through electric motors, it causes them to spin. And this very same thing happens here. This rotor complex actually spins and uses its mechanical energy created by the concentration gradient of the H plus, or electrical energy, turns into mechanical energy, and then is able to take an ADP molecule, which I shall draw in a maybe blue so it'll stand out down here. An ADP comes along and is phosphorylated into ATP. And that's where the energy is then transferred. I guess what we should do is we should say ADP plus, also there has to be a phosphate somewhere, so plus an inorganic phosphate that's floating by as well. Those two things together, you have to have the phosphate on there. ATP. So it's not that difficult to understand in its basic sort of form. So what do you have to get from this? The hydrogen ion gradient that was produced by the electron transport chain is now used to power this special ATP synthase, which is like a motor. And the pushing, or the hydrogen ions that push back through, and the reason they go back is because of the concentration gradient, right? From high to low. They're following the gradient. They're doing what the universe wants. They're reestablishing the equilibrium. So we have forced the equilibrium out of whack using energy, which means we have increased the energy, but we've lowered the entropy of the entire thing. It's more organized. But now Mother Nature is saying, no, 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 back to entropy. We like disorder. And so those H pluses have to go back and reestablish the equilibrium, so they flow down the concentration gradient. And as they flow, they create an electric current, which transforms this protein into a little ATP making machine. And the energy is then translated into a mechanical motion that actually spins this little thing. And then it actually then is then retranslated into the chemical energy by adding the phosphate onto the ATP. Okay, I'm going to pause for a second. I'm going to look up, there's some cool animations on YouTube. I'm going to see if I can find one here to show it to you. Here it is, ATP synthase in action. So we'll watch this little video. It's only 4 or 5 minutes long, and see if we can't get a better appreciation for what's actually going on here. I'm going to pause for a second because he's going to be talking about all of the different pieces and parts, which all have names, which we didn't obviously focus on. So when you hear all these cool, weird names, that's what he's basically looking at. But here you can clearly see the stalk down below. You can see the sort of different proteins that make up that top part. And you can see this stator, this connector blue thing. What you can't see is what's tipping into the membrane. So this is actually an upside down version of what we drew. The part in the membrane is on this side. So his whole picture is from the opposite angle. So this is the small part hanging down the bottom on our picture. It's upside down. Okay, so let's go. Oh, it's delaying, sorry.
We'll now look at a rendering of the structure. Data structures. ATP and inorganic phosphate. The ATP has just simultaneously we zoom into the active site. You can see the molecular structure of the substrate. all held in place by a network of hydrogen bonds, mediated by the unfolding structure of the
So what he's talking about here is how enzymes work, right? The enzymes have a special area, and by enzyme, he's talking about the ATP synthase, which is a giant protein molecule. And so these ADP, which is this green thing, is actually lodging into place and held there by the hydrogen bonds because of the shapes of these molecules, or this enzyme activity. He's talking about the active site, right? So, and then it's going to bring in the phosphate and then stick them together.
in a particular direction. Just the rotation happens in a specific direction. If the water was to flow in the opposite direction, you would see rotation of the water wheel in the opposite direction. Here we see the C ring and the A subunit. We don't know the full structure of the A subunit, but we know that there's an access channel for protons on the A subunit right in the intermembrane space. And there's a separate egress channel for protons on the matrix side. In this animation, you see protons flowing out from an interface between the C ring and the A subunit. But what drives the specific direction of this rotation? For example, if we were looking down, it is always a counterclockwise rotation. Why and what drives that? On each of the C subunits within the C ring, there is a proton binding site. Notice the high concentration of protons on the intermembrane side and their flow to the lower concentration on the matrix side. Protons load from the highly concentrated intermembrane side onto the C subunits, and they are released on the matrix side where the concentration of the protons is low. But why can't protons go around the other way as well? In principle, just as in the water wheel analogy, the C ring should be able to rotate in either direction, shouldn't it? There are two interconnected reasons for this unidirectional movement. The amino acid side chains in C subunits would repel the bound proton, so that when a proton loads from the intermembrane side, the C ring cannot rotate back in the opposite direction because it is energetically unfavorable. Rotating counterclockwise, on the other hand, is energetically favorable because there are no repulsive interactions. Remember, there's a concentration gradient that favors binding of protons on the intermembrane side and the release of protons on the matrix side. So in summary, the proton gradient determines the direction of the rotation, favoring movement in the counterclockwise direction of the C ring. This avoids unfavorable interactions with the amino acid side chains of the C ring.
Yeah, that's worth actually. The question was asked while we were paused about proton, and remember that a proton is just another name for an H plus ion. A hydrogen ion, stripped of its electron, is essentially just its nucleus, which is a proton. So that's what they meant by that. Let's see if I can't get back now. Okay, so we're back to our picture. I apologize when we played that video on, if you're watching this in video form, the sound may not be very good because I realized it wasn't coming through my system, it was coming through the projector. So you may have to go to YouTube, find that video, go there and watch it if you're watching this at home, because you won't be able to hear what they're saying in the video. But again, they said a lot of things that are beyond what we're talking about. They were talking about the subunits, which are these blue sausage-shaped proteins. But they did show you, which was nice, the concept of how this functions as an enzyme with an active site that actually is able to hold on to an ADP. So when we draw that ADP at the bottom there coming in, what we mean is it's attaching to its active site. The phosphate is attaching to an active site, and the protein is using its mechanical energy to force them together into a bond, so to speak. Okay, so that's how the ATP synthase works. Any questions on that before we go into a sort of overall summary? Okay, so what we'll do, we'll just continue this, and we're going to do an overall summary of the entire process. Just looking at the production of energy. And we'll do this in a sort of a flowchart form. Overall summary. I'm going to use a thinner line here. So essentially, we take a glucose molecule, and that glucose molecule is basically turned into two pyruvate molecules. Maybe, yeah, you know what? I'll just use a number 2. I think the picture will get too complicated if I try drawing everything with double arrows. So that gets turned into 2 pyruvate molecules, 2 pyruvate. And of course, that is the process of glycolysis. Okay? So if you wanted to write here, glycolysis. Oh, shoot. I wish it would remember what size I like to use. So if you wanted to write on here, glycolysis, there. That's the process. And what we need to remember is basically what comes out of that. So I got my 2 ATP go in, right? I'll put in blue the fact that 2 ATP are used to sort of go into that and charge that process, get it going like the match that lights a fire, but then 4 ATP are produced in the process. So the net gain, as we said, was 2 ATP. The other thing that we get are 2 NADHs that come out of this as well. So in addition to that, another arrow, small 2 NADH. And that's because the NADH is produced after the molecule splits into 2. So there's 1 produced at that step, but because everything is happening twice, after the glucose splits into 2 molecules, then we end up with getting 2 NADH out of that process. And there are other things that happen, but this is going to be just tracking the energy-carrying molecules only, okay? So then the 2 pyruvate get turned into something different. They get turned into 2 acetyl-CoAs. So I'll write those down here. Acetyl-CoA. I'll just write it like that. And there's some stuff that happens here in terms of energy as well. We get the carbon dioxide. Well, I'm going to write the carbon dioxide in. I'll do it in a purpley color, just because that's part of the overall equation. So we'll get coming out of this process carbon dioxide. And actually, there are 2 of them, I believe, because this happens twice. Everything's going to happen twice after this point, right? 2 carbon dioxides are produced as the pyruvate crosses the mitochondrial membrane. I'm going to do this. I'm going to get a crayon, and I'm going to take this yellowy color, and I'm going to put a little sort of barrier here, like this, to remind us that this is the mitochondrial membrane. And in fact, maybe I'll put 2, the inner and outer membrane. There. So remember that now everything that happens is happening inside the mitochondria. You can label that mitochondrial membrane if you want, but I'm not going to because it's going to mess up this picture. I don't have the space. So back to our pen. The other thing, of course, that comes out of this process is 2 NADHs as well. So we'll put that in green. keeping track, comes out of there 2 NADHs. What's interesting about this, and this is a time to point this out, and this is also a good time to practice taking notes from what I'm saying rather than what I'm drawing. But you see the 2 NADHs produced in glycolysis. They cannot enter into the Krebs cycle process unless they cross the mitochondrial membrane. And so they have to also be transported across the membrane. They're high energy electron carriers, and they don't cross the membrane very easily on their own. And so it requires some energy to make that happen. So there are these little hidden energy inputs, like matches, that we need to light every so often. So to get the 2 NADHs from glycolysis on the left side into this picture so they can work on the Krebs cycle, there has to be some active transport there as well. And this is one of the reasons why it's difficult for us to determine exactly how many ATP we're going to make. We're going to have a range, and it's because it depends on the mode of transport for those 2 NADHs to get inside the mitochondria as well. And of course, even the pyruvate, which has to be transported actively into the mitochondria, also requires a transport mechanism that's active and uses up some energy. I'm not going to write all that, but it's an interesting thing to kind of make note of, okay? Because we'll come back to it later. Okay, let's move down. I don't think there's anything else of interest. There is the CoA that goes in and out, but we're just looking at the energy-carrying molecules and the carbon dioxide. So what I'll do down below here is rewrite the 2 acetyl-CoAs. And then for them, what they do is they hook up with citrate, which is sort of well, to be honest, they don't hook up with citrate. They hook up with oxaloacetate and become citrate, which then goes through the Krebs cycle. Okay, so we can put Krebs in here, or the citric acid cycle. There we go. And as long as you keep feeding acetyl-CoAs, this circle keeps on going. So at that point, there's no sort of product that comes out of the black. The black is now trapped in the circle, but there are things that come out of the process. In particular, if we look at our ATPs first, out of this process, we get, we get ATP. Now remember, the ATP comes out once in the cycle, but the cycle is happening twice. So we should probably put a ×2 somewhere on that cycle. Right? 2 acetyl-CoAs will give us 2 citrates, 2 Krebs cycles, right? And that will give us then 2 ATP as a result of that process. We will also get 2 FADH2s. So it's an electron carrier, so I'll put it in. I'll put it in green. And we will also get we'll get some NADs, but we'll end up getting 6 NADs. So out of this process, we get 6 NADHs, actually, NADHs. And if we're keeping track, we also get some more carbon dioxide, which I was writing in purple, I guess. So we can put that in here too as well. That's the rest of the carbon dioxide. We already had 2, and this gives us 4, so that's a total of 6. There's also some interactions with water, a couple other things. We didn't worry about the GTP part because that's another cyclic thing. It produces the ATP. Those are the main things that we want to sort of glean from this Krebs cycle. Okay, so now we need to do an accounting. The ATP is already made, so we'll leave that for the end. But the NADs and FADH2s are the things that have to sort of now go into the electron transport system that we just did. So let's count them all up. All right? So right here. So there are 2 NADHs from glycolysis, And if they're transported across the membrane, they can then also interact with the mitochondria, right? And there's another 2 in the pyruvate oxidation, so that's a total of 4. And then if we go down here, we see 6 more coming out of the double citric acid cycle, which gives us a total of 10. So we've actually produced a total of 10. Let's go back to green. 10 NADH molecules. And the only place we get the FADH2 is in the Krebs cycle, so we have 2 of those. 2 FADH2 molecules. These 2 molecules enter into electron transport, which we'll just write as. So they go through the process of providing the energy to pump the H plus from one side to the other, right? And that process also has a name. It's sometimes called chemiosmosis in your textbook. Osmosis. If you see the word chemiosmosis, it's referring to the part of the electron transport that is the ATP. So electron transport and chemiosmosis, we'll call it. Chemiosmosis is the term used to describe the ATP synthase action, and I didn't mention that before in the video, but if you see that word, that's sometimes referring to just the ATP part of it, the synthase part, the last step. So those 2 are often written like that. And out of this, we get somewhere between 32, 34 ATP. So that results in somewhere between 32 to 34 ATP. And don't forget, we had some ATP before. We had 2 produced from the citric acid cycle, and we had a net gain of 2 from glycolysis. So there's an additional 4 ATP that were already produced. So when you add that together, okay, so when you put all the ATPs together, the total grand ATP count is basically, I won't put an arrow because that looks like a reaction. The total ATP count then, I'll just put a dash, is 36 to 38 ATP. 36 to 38 ATP. So you can see that last part in the mitochondria. You can see why the mitochondrion is such an evolutionary advantage to a cell, because it boosts the energy production like crazy. We go from 4 measly ATPs to being able to produce 36 to 38 because of that membrane and all that stuff that's happening in the last sort of stages. And of course, you will recall that the mitochondrion is believed to have once been a free living organism, like a bacterial cell of sorts. Not really a bacteria because it has inner membrane structure, but it would have been a free living single-celled structure that had developed this membrane system and was using it to power. It probably was a very active thing that could swim around quite readily. But it would very likely become engulfed by larger cells through phagocytosis, where they wrap their membranes around. And of course, over time, a symbiotic relationship could develop between these cells, where the big cell, which swallowed the mitochondria, instead of digesting it and breaking it down, was able to sort of utilize its tremendous energy-making ability, and that set the stage for very high-energy eukaryotic cells, which eventually could produce us and other complex life. So it's kind of a neat thing. Without the mitochondria, we're stuck with 4 lousy ATP, right? Not very much. You can't build skyscrapers with 4 ATP. So there is the sort of last little tally. We should make a note of why the range. Why is it not precise? Why do we have a range here? And I already alluded to that, and the range is because there's probably about 3 different reasons. One reason is that there is some active transport of NAD H and pyruvate across the mitochondrial membranes to get them inside. These take energy, and the pyruvate is really not so much of a problem, but the NAD is, because it's unclear exactly how much ATP it takes to transport that. It can be transported by more than one mechanism, and the mechanisms each use different amounts of energy, and so doing a sort of net total means it's a little bit fuzzy in terms of that. The other thing that can happen is that there are other things are making use of that proton gradient. Other processes in the cell may make use of the proton gradient, the hydrogen ion gradient that's been made. So yes, it's flowing through the ATP synthase making ATP, but that hydrogen ion concentration might be forcing those H pluses through different proteins in the membrane that are doing other biochemical reactions. So other processes may make use of the hydrogen ion gradient. Question? Want to go back up a bit? Oh, so I'll just finish by saying, therefore, the exact ATP count is unknown. Essentially, we have 36 to 38, right? 36 to 38 is the best we can do, but it's still a lot. It's still enough to power cells substantially to do the things that they do. Okay, that's the end of that. So we'll stop there.